The virus is an infectious particle that exhibits the characteristics of life and non-life. The structure and function of the virus differs from plants, animals, and bacteria. In this lecture, we're going to spend some time just talking about the special and very unusual properties of viruses. First, we need to consider the enormous amount of biological diversity that we see within the viruses. There's more diversity within viruses than in all the rest of the bacterial, plant, and animal kingdoms put together. If you stop and think about it, this makes sense. This diversity results from the success of viruses in parasitizing all known groups of living organisms. So basically, every organism has at least one virus that infects it. Understanding this diversity is the key to comprehending the interactions of viruses with their hosts. Viruses are typically described as obligate intracellular parasites, acellular infectious agents that require the presence of a host cell in order to multiply. Viruses replicate in a way completely different from other organisms. There's no mitosis or binary fission. Instead, virus particles are produced from the assembly of preformed components, whereas other agents grow from an increase in the integrated sum of their components and reproduce by division. In other words, virus replication is similar to producing cars in a factory. Also, viruses are energy parasites. Viruses lack the genetic information which encodes the apparatus necessary for the generation of metabolic energy or for protein synthesis. Viruses have neither mitochondria nor ribosomes. They are completely dependent on a living host for their survival. This explains why early researchers had so much trouble trying to grow viruses. Unlike bacteria, which merely required an appropriate medium, viruses required living hosts or cells. Viruses, if you recall from our first lecture, were originally set apart because of their small size. Remember, they were originally called filterable agents. A common mistake is that viruses are always smaller than bacteria. While this is true in most cases, size alone does not serve to distinguish between them. The largest virus particles, the granuloviruses, are almost 300 nanometers in diameter, and some are as much as 500 nanometers long, while the smallest bacteria, for instance some of the mycoplasmas, are only 200 to 300 nanometers long. So size alone does not differentiate between viruses and bacteria. Viruses can be extremely simple in design, consisting of nucleic acid surrounded by a protein coat known as a capsid. In fact, virologists often joke that viruses are bad news wrapped in protein. The capsid is composed of smaller protein components referred to as capsimers. The capsid plus genome combination is called a nucleocapsid. Viruses can also possess additional components, with the most common being an additional membranous layer that surrounds the nucleocapsid, called an envelope. The envelope is actually acquired from the nuclear plasma membrane of the infected host cell and then modified with viral proteins called peplimers. Some viruses contain viral enzymes that are necessary for infection of a host cell and coded for within the viral genome. A complete virus with all the components needed for host cell infection is referred to as a virion. In the next series of slides, we're going to look at these different aspects of virus morphology in more detail. Viral nucleocapsids come in two basic shapes, although the overall appearance of a virus can be altered by the presence of an envelope. Helical viruses have an elongated tube-like structure with the capsimers arranged helically around the coiled genome. Icosahedral viruses have a spherical shape, with icosahedral symmetry consisting of 20 triangular faces. The simplest icosahedral capsid has 3 capsimers per triangular face, resulting in 60 capsimers for the entire virus. Some viruses do not neatly fit into either of the 2 previous categories because they are so unusual in design or components. So there's a third category known as complex viruses. Examples include the pox virus with a brick-shaped exterior and a complicated internal structure, as well as the bacteriophage with tail fibers attached to an icosahedral head. The capsid serves a really important function for a virus. It protects the fragile nucleic acid genome from physical, chemical, or enzymatic damage. Nucleic acids are susceptible to physical damage such as shearing by mechanical forces and to chemical modification by ultraviolet light, such as from sunlight. The natural environment is heavily laden with nucleases, either derived from dead or leaky cells, or deliberately secreted by vertebrates as defense against infection. The protein subunits in a virus capsid are redundant. That is, they're present in many copies per particle. Damage to one or more subunits may render that particular subunit non-functional, but usually it does not destroy the infectivity of the whole particle. The outer surface of the virus is also responsible for recognition and the first interaction with the host cell. Initially, this takes the form of binding of a specific virus attachment protein to a cellular receptor molecule. The capsid also has a role to play in initiating infection by delivering the genome in a form which can interact with the host cell. In 1957, Frankel, Conrad, and Williams showed that when mixtures of purified tobacco mosaic virus RNA and the virus's coat protein were incubated together, virus particles formed spontaneously. This indicated that the particle was in the free energy minimum state and was therefore the favored structure of the components. In other words, the virus particle would self-assemble. Stability is an important feature of virus particles. Although some viruses are very fragile and unable to survive outside the protected host cell environment, many are able to persist for long periods of time in the environment. Helical capsids are composed of a single type of subunit stacked around the nucleic acid, forming a central axis. This arrangement results in rod-shaped or filamentous virions. They can be anything from short and highly rigid to long and very flexible. Most often, the genetic material is single-stranded RNA. The structure is kind of like a spiral staircase. It's also a very stable structure. Each subunit forming the capsid forms bonds with the subunits next to it and the subunits in the turn before and the turn after it. This is the simplest way to arrange multiple identical protein subunits. And many important viral pathogens have this kind of symmetry. You can see some of the varieties that are possible in this picture. So for instance, tobacco mosaic virus down in the bottom right corner. is just a naked capsid, so you have just these long, almost stick-like nucleocapsids. On the left at the bottom is an example of rabies virus, an electron micrograph, and it's a single capsid with an envelope around it. And then many of the paramyxoviruses, like you see at the top right, will have multiple capsids within a single envelope. Many different plant viruses show helical symmetry. They'll also be naked. They don't have an envelope. It's unknown why so many different plant viruses have evolved this structure, but it may be related to either the biology of the host plant cell or alternatively to the way in which they are transmitted between hosts. Remember, a plant cell is very different from a host cell, particularly because of the cell wall around the plant cell. And that's a major obstacle that a virus particle has to be able to cross to infect a plant cell. Now we see many examples of helical animal viruses as well. Some really important pathogens fall into this group, for instance, rabies. But one thing that's interesting to note is that there are no naked helical animal viruses. They just don't exist. All helical animal viruses have an envelope. And as I mentioned, there are many important pathogens in this group, including influenza, mumps, measles, and rabies. Icosahedral capsid symmetry results in a spherical appearance of viruses at low magnification, but actually consists of multiple copies of proteins called capsimers arranged in a regular geometrical pattern similar to a soccer ball. Hence, they are not truly spherical. Icosahedral architecture was employed by R. Buckminster Fuller in his geodesic dome, and this is the most efficient symmetrical arrangement that subunits can take to form a shell with a maximum internal capacity. Most animal viruses have icosahedral symmetry. The basic icosahedron is a solid shape consisting of 20 triangular faces arranged around the surface of a sphere. So let's look at icosahedral symmetry a little bit closer. So as I said, the simplest icosahedron has 20 faces. Each is an equilateral triangle. You have to have 20 subunits to make an icosahedron. If you take 19 triangles and try to put it together, you're going to be missing a spot. That's just the geometry of the structure. You have to have 20 faces. Each of those faces, each triangle, is formed from 3 protein subunits, or capsimers. Again, that's the geometry. There has to be 3. And together, these allow the formation of a shell with the smallest number of identical subunits, in this case 60. So you have 20 faces with 3 protein subunits forming each face, so 3 times 20 is 60. Now when you look at the arrangement of these faces, you'll see that there's various axes of symmetry. At the vertices, you have 5 of these triangles coming together, and it's a 5-fold axis. You can see that indicated by the green. Also, though, each triangular face interacts with 3 others, forming a 3-fold symmetry, and then you have a 2-fold symmetry. Let's look at this a little more closely. So here we have just another way of illustrating this. So we have our icosahedron. It has 60 identical protein subunits. Those commas on the figure are to indicate protein subunits. Remember, it's the protein subunit, that's the structural unit of this. That's what is used to put the whole thing together. And this just further indicates that 2, 3, 5 symmetry that we talked about. So at the blue spot where the heads come together, that's twofold symmetry. The red triangle, that's where the necks come together, that's threefold symmetry. And then at the vertices, you have fivefold symmetry. That's where the tails come together. So again, this is our simplest icosahedron. You use 3 identical subunits to form each triangular face, and you have 20 triangular faces. This is what we call t equals 1 in terms of our triangulation number. And this works for a lot of viruses, but not all viruses are the same size. So the question becomes, how do you make bigger viruses? So if a virus with a triangulation number of 1 is the simplest icosahedron, it's also the smallest. We can make larger icosahedrons, but we can't do that by just making the protein subunits larger. There are biological limits to the size of proteins. The general rule of thumb for the length of a typical protein in prokaryotes is about 300 amino acids, and in eukaryotes it's about 400 amino acids. Therefore, making the subunits larger isn't possible. Instead, you increase the number of subunits. Now, the 3 types of contact between the subunits remains the same. So you have head-to-head for the twofold, back-to-back for the threefold, and then you have tail-to-tail. But in this case, as the virus gets larger, you can add another kind of symmetry. We'll look at that in the next slide. But here you've got 2 viruses, t equals 4 and t equals 3. Each triangular face is made up of 3 capsimers, but we have more of the triangular faces. And notice that the numbers 240, 180, those are all multiples of 60. So to make your icosahedral bigger, you increase the number of capsid proteins, but your constraints by the geometry of the icosahedral symmetry doesn't change. So you're always going to need your 60 capsid proteins per unit size. So as at t equals 1 you have 60, at t equals 3 you have 160. As the size of the virus gets larger, you can, again, you're going to be increasing the number of subunits. You're going to have more triangular faces. And what you'll find is with the larger capsids, you increase from 5-fold symmetry to areas where you have 6-fold axis of symmetry. And you can see that here in this picture. And again, the 5-fold or 6-fold axes of symmetry are going to be at the vertices of the icosahedron. It's where you have the tail-to-tail contact between the subunits. And here's just an illustration of 3 different viruses. We have t equals 1, t equals 3, and t equals 4. And you can see that this allows the virus size to be larger. The icosahedron becomes larger. And we've increased the size of the icosahedron by increasing the number of subunits. So t equals 1, remember that's the simplest. So you have 60 subunits. You go to t equals 3, and you have to have 180 subunits, 3 per triangular face, and 180 is merely 60 times 3. That's your triangulation number, 3. And with t equals 4, the number of subunits is 240, so 60 times 4. Many virus particles are naked. That is, they have the capsid proteins exposed to the external environment. These viruses are usually produced by lysis of the infected cell. That's how they're released into the environment. Obviously, though, this process results in the death of the cell. Many viruses have devised strategies to exit from the infected cell without its total destruction. They do this by a process called budding. As the virus buds through a cell membrane, it acquires an envelope. The envelope consists of a phospholipid bilayer with a mixture of viral proteins and proteins derived from the plasma membrane of the host cell in which the virus was manufactured. The presence or absence of a lipid envelope is another important defining property of different groups of animal viruses. This is another look at the process of budding. This is achieved by extrusion or budding of the particle through the membrane of the host cell, during which process the particle becomes coated in a lipid envelope derived from the host cell membrane and has a similar composition to the host cell membrane. Should be kept in mind, though, that although many viruses bud from the plasma membrane, they might bud from other membranes in the host cell as well. So, for instance, herpes virus replicates in the nucleus of the host cell and buds through the nuclear membrane. Some viruses bud through the membranes of the ER, so it depends on the particular virus. The envelope provides additional protection from desiccation and enzymatic damage. We already talked about how the capsid is protective. Having an envelope provides additional protection as well. But remember how the envelope is acquired. It's acquired by the virus as it buds through a cell membrane. And so it's derived from these host cell membranes. It's going to have various proteins and carbohydrates and things that are present in the host cell membrane in the envelope. But when we look more closely, we find that in addition to cellular components, we also find viral encoded proteins in the envelope. And there are different types of envelope proteins. There are the matrix proteins, which are internal proteins. Their function is to effectively link the internal nucleocapsid assembly to the envelope. Such proteins are usually not glycosylated and are often very abundant. For example, in retroviruses like HIV, they comprise approximately 30% of the total weight of the virion. However, on the outside, we find glycoproteins. These are transmembrane proteins anchored to the membrane of a hydrophobic domain and can be subdivided into two types by their functions. External glycoproteins are anchored in the envelope by a single transmembrane domain, and transport channel proteins contain multiple hydrophobic transmembrane domains, forming a protein-lined channel through the envelope. Glycoproteins, these more external envelope proteins, are really important in terms of virus functioning. They play a role in virus structure, of course, but they're also key in virus attachment. How does a virus attach to a host cell? Well, the key to that interaction are these envelope proteins. Envelope proteins often have a fusion activity, so that when the virus binds with a host cell, the envelope protein will trigger a fusion event so that the capsid or the nucleocapsid can enter into the host cell. Another aspect of glycoproteins, because they are on the surface of the virus and they play a role in virus attachment, they're primary targets of the immune system. Complex viruses possess a capsid which is neither purely helical nor purely icosahedral, and which may possess extra structures such as protein tails or an outer wall. For instance, poxvirus particles are the most complex virions known and contain more than 100 virus-encoded proteins arranged in a variety of internal and external structures. Bacteriophages are microscopic particles with a head and tail. The head contains viral nucleic acid. Some phages have tails and some have tail fibers. Bacteriophages attach to bacteria using their tail or tail fibers. Then they inject their nucleic acid into the bacterial cell. There the virus multiplies. Finally, the newly produced virus bursts from the bacterial cell, killing the bacterium. This releases the virus into the host's environment. But notice in the sequence at the bottom of the slide, the various component parts of the bacteriophage are produced separately and then assembled into individual viruses. So think back to what we were talking about in regards to virus replication. The Gemini virus consists of two twinned T=1 icosahedra. Each icosahedron has one morphological subunit missing, and the icosahedra are joined at that point, such that the mature particle contains 110 protein monomers arranged in 22 morphological subunits. The icosahedron for Rheoviridae is T=13. You have capsids composed forming a double protein shell with a very complex structure. Most viruses only have a single protein shell. So this is one way that the reoviruses are very different from other viruses. The baculoviruses are natural pathogens of arthropods and are used as biocontrols. Some baculovirus particles exist in two forms: a relatively simple budded form found within the host insect, and a crystalline protein occluded form that's responsible for environmental persistence. This illustration just shows you the variety of structures that we see in virus particles. So again, we have the helical type virus, we have the icosahedron, we have enveloped viruses, and then of course we have the complex viruses. The different virus morphologies we have discussed do not occur with equal frequency among animal, plant, and bacterial viruses. There are relatively few purely icosahedral viruses in bacteria. Non-enveloped viruses are common and occur almost exclusively in plants. Enveloped icosahedral viruses and enveloped helical viruses are common in animals and rare in plants and bacteria. Finally, the complex head-to-tail morphology is seen only in bacteriophages. Unfortunately, there's no real explanation as to why these restrictions exist. There are also some very large, very complex viruses whose morphogenesis is beyond our current comprehension. Viruses can be classified on the basis of their structure, but also on the basis of their viral nucleic acids. DNA is the hereditary material in all cells. As cells synthesize the molecules they need to function, information flows from DNA to messenger RNA to proteins. Although all cells follow this pattern, which is a central dogma of molecular biology, most viruses break it. In fact, viruses demonstrate an amazing diversity of genome types, and this is used in part to classify viruses. Also, the number of genes found in viruses varies widely. The thymoviruses that infect plants contain as few as 3 genes, but the genome of smallpox can code for up to 340 proteins. But we can use the Baltimore scheme of virus classification, and this is based on the nucleic acid. So the first question is it RNA or DNA? Many viruses will use RNA as the genetic material rather than DNA. Then we have to ask, is it single-stranded or double-stranded? We're accustomed to think of DNA as existing as a double-stranded molecule and RNA as a single-stranded molecule. But in viruses, again, they break the rules. There are viruses that will have a double-stranded RNA genome, and there are some that have a single-stranded DNA genome. And then we have to consider whether it's segmented or non-segmented. Some viruses will have just a single piece of nucleic acid forming the genome, but then other viruses will have multiple segments, sort of akin to chromosomes. Then, are these nucleic acids linear or circular? Many will have a linear formation, but some are circular. And then, is it haploid or diploid? With many eukaryotic organisms, of course, we think of diploid or double copy of the chromosomes. Most viruses are what we would consider haploid, but there are some exceptions. But of course, the nature of the nucleic acid is going to determine how the virus replicates. And we'll be going into this in much more detail later in the course. So here you can see the kinds of virus classifications that we have. So the first level of classification would be nucleic acid. Is it RNA or DNA? So that divides the viruses into 2 major groups. Then within each group, is it icosahedral or helical? Then we go down to naked or enveloped. And then we can look at genome architecture and the Baltimore classification scheme. So this is how we set out to classify viruses. And you can see some of the more common viruses listed down below. Besides viruses, there's some other very small virus-like particles that are infectious and cause significant disease, but they don't really fit into any other kind of category. These would be the viroids, the virusoids, and the prions. So on this slide, I've given you the basic definitions of them for a quick comparison, but let's look at them in more detail. So first are the viroids. These are small, single-stranded, circular, non-encapsulated, pathogenic RNA molecules that are important infectious agents in plants. They do not code for proteins and thus depend on plant host enzymes for their replication and other functions. It is unclear how viroids cause disease. Viroids can be spread from plant to plant through mechanical damage caused by insects or by cultivation. They are also spread by propagation of cutting from infected plants. Viroids can also be present in seed. Very often they're transmitted during the manipulation of crop plants for harvest. And here's some illustrations of viroid diseases. More than 20 viroids have been described infecting a wide variety of plant species. Many of these have great agricultural significance and are known to destroy fields of economically important crops. Virusoids are satellite, viroid-like molecules, somewhat larger than the viroids, which are dependent on the presence of virus replication for multiplication. That's why we call them a satellite. And they're packaged into a virus as passengers. So in this case, we have hepatitis D. That is a virusoid, and it's a satellite. It cannot replicate in the absence of hepatitis B virus, which is a fully functional infectious virus. So in this case, hepatitis D is a satellite, and it requires hepatitis B as a helper virus. We'll talk more about that when we get into to hepatitis B. Prion diseases, or transmissible spongiform encephalopathies, are a family of rare, progressive neurodegenerative disorders that affect both humans and animals. They are distinguished by long incubation periods, characteristic spongiform changes in the brain, and a failure to induce inflammatory response. A prion is an abnormal transmissible agent that is able to induce abnormal folding of normal cellular prion proteins in the brain, leading to brain damage and the characteristic signs and symptoms of the disease. Prion diseases are usually rapidly progressive and always fatal. And there are some well-known examples of this. For instance, CJD, or Creutzfeldt-Jakob disease, and of course bovine spongiform encephalopathy, or BSE, and chronic wasting disease, or CWD. Creutzfeldt-Jakob disease, or CJD, is a rare degenerative, invariably fatal brain disorder. It affects about 1 person in every 1 million people per year worldwide. In the United States, there are about 300 cases per year. CJD usually appears in later life and runs a rapid course. Typically, onset of symptoms occurs about age 60, and about 90% of individuals die within 1 year. In the early stages of disease, people may have failing memory, behavioral changes, lack of coordination, and visual disturbances. As the illness progresses, mental deterioration becomes pronounced, and involuntary movements, blindness, weakness of extremities, and coma may occur. BSE is a progressive neurological disorder of cattle that results from infection by a prion. Research indicates that the first probable infections of BSE in cows occurred during the 1970s, with 2 cases of BSE being identified in 1986. BSE probably originated as a result of feeding cattle meat and bone meal that contained BSE-infected products from a spontaneously occurring case of BSE or scrapie-infected sheep products. Scrapie is a prion disease of sheep. There is strong evidence and general agreement that the outbreak was then amplified and spread throughout the United Kingdom cattle industry by feeding rendered prion-infected bovine meat and bone meal to young calves. Strong epidemiologic and laboratory evidence exists for a causal association between a new human prion disease called variant CJD that was first reported from the United Kingdom in 1996 and the BSE outbreak in cattle. The interval between the most likely period for the initial extended exposure of the population to potentially BSE-contaminated food and the onset of initial variant CJD diseases is consistent with known incubation periods for the forms of prion disease. The U.S. Food and Drug Administration is doing many things to keep the food in the U.S. safe for both people and cows. Since August 1997, the FDA has not allowed most parts from cows and certain other animals to be used to make food that is fed to cows. This protects healthy cows from getting BSE by making sure that the food they eat is not contaminated with the abnormal prion. In April 2009, the FDA took additional steps to make sure that the food in the U.S. stays safe. Certain high-risk cow parts are not allowed to be used to make any animal feed, including pet food. This prevents all animal feed from being accidentally contaminated with the abnormal prion. High-risk cow parts are those parts of the cow that have the highest chance of being infected with the abnormal prion, such as the brains and spinal cords from cows that are 30 months of age or older. The FDA also works with the USDA to keep cows in the U.S. healthy and free of BSE. The USDA prevents high-risk cows and cow products from entering the U.S. from other countries. The USDA also makes sure that high-risk cow parts, such as brains and spinal cords, and cows that are unable to walk or show other signs of disease, are not used to make food for people. The steps that the FDA and the USDA have taken to prevent cows in the U.S. from getting BSE are working very well. Only 6 cows with BSE have been found in the U.S. The first case was reported in 2003, and the most recent case was found in August 2018.